everyone. I'm going to tell you about my research into the temporal and spatial variability in benthic microalgae in a shallow coastal lagoon. First, what is benthic microalgae? Benthic refers to anything that is in the bottom layer of a body of water, and microalgae is microscopic algae. So, benthic microalgae is microscopic algae that lives at the bottom of a body of water. And why is benthic microalgae important? First, it regulates the cycling of nutrients like nitrogen and phosphorus, so it ensures that those nutrients are available in the sediment so that plants can use them to grow. It also stabilizes sediment with extracellular polysaccharides. These are slimy substances that the microalgae excretes that sticks together sediment, so it is not washed up by the tides and the currents and suspended in the water column. This helps keep the water clear so that seagrass can use the sunlight to photosynthesize. Now, the seagrass that grows in the lagoons where I did my research is the specific species eelgrass. And eelgrass creates what we call a positive feedback loop, which means that the seagrass roots help stabilize the sediment, and the blades of the seagrass help slow down the movement of water. And these things also prevent sediment from being suspended in the water. So the more seagrass there is, the better the conditions are for more seagrass to grow. But if this cycle is broken, it's hard for the seagrass to reestablish. Seagrass is also important because it creates food and habitat for many organisms that live in the lagoons. So, the questions that my research focused on are: does increased temperature impact the quantity of benthic microalgae? Does increased temperature impact the biodiversity of benthic microalgae? And how do these factors vary between lagoons? I did my research at the Virginia Coast Reserve. This is an area off the eastern shore of Virginia consisting of different lagoons and barrier islands that are protected for conservation and ecological research. In the 1930s, there was a die-off of almost all the eelgrass in this area, which was caused by a disease that was going through the seagrass community and a large storm. In the 1990s, there was a project that was started to replant the seagrass meadows, and this went well until 2015, when a particularly warm summer caused dieback of the seagrass. Since then, the seagrass has been growing back, but this raises concerns about warmer temperatures of the water as the climate continues to change. These are the sites where I did my research. I chose these sites because they have different levels of heat stress and different seagrass densities. This channel here is where cooler ocean water can wash in with the tides. So the sites close to this channel have cooler temperatures, and the sites further away from that channel have warmer temperatures and are more heat stressed. In each bay, I chose 3 sites, one where the seagrass is more dense, one where the seagrass is sparse, and one that is mostly bare of seagrass. At each site, I placed one of these sensors that recorded the temperature and the light that reached the sensor every 15 minutes. Each week, I visited each site to take 10 1-centimeter sediment cores, which are samples of the sediment that go 1 centimeter deep, and 1 surface sample that skims the top of the sediment and the very bottom of the water. Here are my preliminary results. I graphed the chlorophyll A, which is a measure of how much microalgae there is, versus the temperature in degrees Celsius. As you can see, the data is a bit messy, but this is what we would expect from a complicated system where we not only have to take into account the temperature, but also the density of seagrass, what nutrients are in the sediment, the grain size of the sediment, what other organisms are in the area, and a variety of other factors. I tried separating the data out into the different seagrass density categories. So here's the data for the dense sites. As you can see, there is a bit of an increasing trend between chlorophyll A and temperature, but the data is still pretty variable and it is hard to tell for sure. This is much the same with the sparse sites. Again, at the low chlorophyll A measurements are also found where there are lower temperatures, but then this data spreads out at the higher temperatures. And for the bare sites, it's hard to see what the correlation may be. I have started some further research to answer more of my questions. First, I've been looking at a new method for analyzing my data. There has been some lab research that shows that there is a threshold temperature above which the benthic microalgae is less productive. So I've tried looking at how much time in the week preceding sampling the microalgae was above this threshold temperature. I have also worked on some lab replication of this experiment, and I've also looked at identifying the diatoms, which are the different kinds of microalgae. First, for the data analysis, you can see the time versus temperature at two of my sites. As you can see, they follow approximately the same trends, but one site is more heat stressed than the other. Some research has showed that above 24 degrees Celsius, so approximately this line, is where benthic microalgae is stressed. So I will look at how much time the sites are above that temperature and compare that to the amount of chlorophyll. During the fall 2019 semester, I worked on replicating my experiment in a lab setting. I set up 4 tanks to be at an unstressed temperature and 4 tanks to be at a stressed temperature. I measured how much benthic microalgae was produced in each of these tanks. This data can be used to verify that benthic microalgae is less productive above a certain temperature. I've also made some microscope slides to look at the organisms and try to identify them. These are a couple examples that are like the other things that I saw. I have not been able to identify these specifically, but I would like to look at a more magnifying microscope to better tell them apart. I would like to thank Dr. Soika for leading this research, the National Science Foundation for funding my work over the summer, and everyone at the Anheuser-Busch Coastal Research Center for helping me collect my data. Thank you!